Virus strain 100% mortality from humanized transgenic laboratory mice. Please pause each illustration for detailed study of this review of the literature. Illustrations of a laboratory mouse and transgenic mice expressing green fluorescent protein, which glows green under blue light. The central mouse is wild type. Use of transgenic humanized laboratory mice allow for the modeling of human disease development under controlled conditions, specific target gene study, testing of novel drug treatments for faster results, and replication of specific disease characteristics, symptoms, and pathology. Scientists in Beijing of the People's Republic of China have reportedly cloned mutant coronavirus strain GX.P2V infected experimental mice. Gene knocking consists of targeted gene insertion at a specific genomic locus, modifying the endogenous genome for the detailed study of specific mutations of small nucleotides in the context of human disease. In genetically humanized mice, a human gene, genomic sequence, or regulatory element is knocked into the mice in tandem with the knockout of the murine gene, replacing it with the human gene. The infected human angiotensin-converting enzyme 2, or ACE2 receptor mice, were genetically engineered to reflect the genetic makeup that in some aspects reflects that of human beings. ACE2 functions as the entry receptor of SARS-CoV-2 into virus host cells. These humanized laboratory mice exhibited a 100% death rate in a remarkably rapid 8 days post-inoculation. The mice experienced rapid weight loss, hunched posture, sluggishness, and complete whitening of the eyes. High viral loads of GX.P2V were discovered in the bones, eyes, lungs, noses, trachea, and most notably the visibly shrunken brains, where infection was of sufficient severity to cause the ultimate death of the experimental mice. Brain involvement has been previously observed in human COVID patients experiencing severe morbidity, despite this disease being largely respiratory in nature. It has been speculated that this cloned mutant virus underwent virulence-enhancing mutation during its time in storage prior to experimental use. It was concluded that this SARS-CoV-2 related pangolin coronavirus is capable of causing 100% mortality in the genetically engineered human angiotensin converting enzyme 2 mice, suggesting a possibility of GX.P2V spillover into the human population. Some scientists have alleged that a gain-of-function study may have resulted, highlighting the necessity of the highest biosafety level of experimental practices and laboratory facility containment when research involving pathogens possessing human pandemic potential are involved. Cartoon of humanized mice model. Refer to nomenclature and abbreviations on the following frame as necessary. Nomenclature of humanized mice and abbreviations. Experimental example of a humanized mouse. Development and characterization of humanized microglial mice. Humanized microglial mice serve as a tool to elucidate neuro-HIV pathobiology and to develop therapeutic and elimination antiretroviral strategies for HIV infections. Interleukin 34 was transgenically introduced, and at the same time, CD34 plus human stem cells were injected into the liver of immunodeficient newborn mice. These mice developed a functional human lymphoid system comprised of B cells, T cells, and macrophages, and brain enriched with human microglial cells. Blue and red-colored cells are human and mouse cells, respectively. In regards to the timing, human cells are introduced after birth to reconstitute lymphoid and solid organ tissues devoid of endogenous murine immunocytes. The human immunocytes show limited number reductions with time after injection as new cells are produced from progenitors. These exist over time less frequently in brain and periphery compared to mouse cells. Reasons underlying restricted species specificity of human pathogenic viruses and solutions provided by the use of transgenic and humanized mouse models. Schematic overview of limitations in the use of mouse models for research of human pathogenic viruses left, and examples of different transgenic humanized mouse models providing solutions to overcome the respective restrictions, right. Associated boxes, limitations, solutions are indicated by the use of the same color. Remaining challenges in the optimization of humanized mice. Four biosafety lab levels. Further determinative and confirmatory investigation, as well as peer review, is indicated corresponding to the research topic of this presentation. New presentations coming soon. Like, share, subscribe, and ring the bell for notifications. Thanks for watching. Thank you to current and new subscribers.